In this section of the PowerPoint, we're kind of still continuing with the cells. So far we've gone over the basic structure of the cell. What is the cell, what is the cell membrane made up of? How does the cell membrane work? And kind of we're working our way inside the cell. This next section, part C, is getting into some of those organelles found inside the cell. So we already know what the cell membrane's made up of. We know how it helps get things in and out, regulating things getting in and out, regulating water getting in and out with osmosis. But now we're to the inside of the cell. We're into various types of organelles. We're into the fluid that's inside of there. We're inside the cell. And so the first part we're going to talk about once we're inside the cell is that fluid. It's the cytoplasm. It's pretty much everything that's inside the cell. It's the liquid. It's any types of let's say insoluble molecules that aren't dissolved in that fluid, and all the organelles that are found inside the cell. So it's pretty much everything inside the cell between the plasma membrane, and it doesn't include the nucleus. Now the cytosol itself is the fluid part of the cell. A lot of times people talk about the cytoplasm as the fluid. It's not technically cytoplasm as the fluid and the organelles and everything else that's in there. But the cytosol itself is just the fluid. It's a water-based fluid, and it does have some soluble molecules, protein, salt, sugars that are all found in there. The inclusions that we can find inside that liquid are insoluble molecules. So they're larger structures, doesn't dissolve in that water-based material, and it's just things we find in there. There's various types of vacuoles, there's some glycogen, there's some pigments, kind of depends on the cell and what kind of inclusions there are. And there's organelles. These are the larger structures found inside the cell. They each have their specialized function, and that's what we're going to focus on these next slides, are those organelles and what are their functions. Now, we do break up our organelles into two groups, those that have membranes and those that don't. And we're going to talk about them. Well, we kind of go back and forth, but the membranes, and these membranes are phospholipid membranes, just like the cell membrane, but they're phospholipid membranes found on the outside of the organelles, and that allows these membranes to have some compartments. They're usually a little bit more higher tech type of organelles that have these membranes. And the first one we're going to talk about is the first one on that list. It is the mitochondria. Now, not only does this organelle have a membrane, it actually has two membranes. It has one on the outside and one on the inside. And I go back and I'm like, so it has an outer membrane, this dark orange, but it also has this folded membrane. It's an inner membrane. And then it has a fluid-like matrix inside of it. Now, all the folds are called a cristae, and their main job is to give lots and lots of surface area. Now, the main function, and I bolded it for a reason, the main function of your mitochondria, it makes ATP. It makes that energy currency that the cell needs to run all of the chemical reactions that happen inside the cell. Now, the most ATP can be made during aerobic cellular respiration, meaning there's oxygen present. Some AT can be made without oxygen, but it's not made in the mitochondria. But I'm like, most ATP is made, lots and lots of ATP is made in the mitochondria when there's oxygen present. Now, it's a pretty high-tech organelle. I mean, it does have two membranes. It even has its own DNA, its own RNA, and its own ribosome. So it's a pretty high-tech organelle all by itself. And yes, its main job is to make energy. It's to make ATP. So sometimes it's called the cell's powerhouse. Sometimes it's called the mighty, mighty mitochondria. But its main job, make ATP. Another organelle, it's non-membranous, are the ribosomes. Their main job, make proteins. We need proteins in lots of areas of the cell. We use them for structural material. We use them for enzymes. I'm like, we use them for recognition. I'm like, there's lots and lots of places we need proteins. That's the job of the ribosome. Now, there's two places we can find ribosomes. One, they're free ribosomes. They're pretty much floating around in that fluid, the cytosol in the cell. And then the other ones are bound, membrane-bound ribosomes. These are the ones that dot that rough endoplasmic reticulum and make it appear rough. Is because of all the little dots of ribosomes all over it. Their still job doesn't matter if they're floating around or if they're stuck to the rough ER. Their main job is to still make proteins. What proteins they make and where they go is a little bit more determined on where the ribosomes are. And so a lot of the proteins that are made on the rough ER, They end up in the cell membrane as maybe transport or carrier proteins. Some end up becoming lysosomes, and some are just exported from the cell to go elsewhere. Now, as we said, ribosomes can be membrane bound, and they can be found on the surface of the endoplasmic reticulum. And so on all of this blue folded material, and even this blue squiggle material, is all endoplasmic reticulum. If it's not dotted with ribosomes, we call it smooth. If it is dotted with ribosomes, we call it rough. Either way, they're right next to that nucleus itself. So the purple is the nucleus, the outside of the nucleus, and you can see these are really close to the nucleus. Here's the nucleus, and again, we've got rough endoplasmic reticulum. All the little black dots are the ribosomes. And on the outside, where you don't see ribosomes, you have some of the smooth ER. Now, because there's rough and smooth, and because some have ribosomes and some don't, their functions are different, even though they're pretty closely found in the same area of the cell. The rough ER, because it has all those ribosomes over it, and the main job of a ribosome is to make proteins, the rough ER, as I kind of italicize words, has a lot to do with the proteins that are made by those ribosomes. So the rough ER manufactures all the secreted proteins, although it is the ribosomes that are doing it on that structure. It helps make some of the integral proteins and some of the phospholipids that are going to become part of the cell membrane. And a lot of these proteins are going to go to the inside of the endoplasmic reticulum. They're going to go into a vesicle, and they're going to get shipped to our Golgi apparatus, which we're going to talk about shortly. So a lot of them get assembled there, made there, and then shipped off to wherever they need to go. Now the smooth ER, which has absolutely no ribosomes whatsoever, so has no function with proteins whatsoever. Instead, it's got a lot of different, very unique functions. A lot of them seem to be lipid driven. And so it does some lipid metabolism, breaking down and making different types of lipids. And it can help make cholesterol, it can help make some steroid-based hormones. It can make lipoproteins, it can help with absorption, synthesis, and even moving of fats, which are lipids. Now, non-lipid type of functions, it can detoxify some drugs out of the body, some pesticides, and even some cancer-causing carcinogenic chemicals. It can also convert glycogen to free glucose, so you've got readily, easily available sugar, and it also can do some storage and release of calcium in the body. So a lot of lipid things, but also some other unique functions as well. So it's got, its own unique characteristic, but nothing to do with proteins. Now, there's the smooth up there. The Golgi apparatus. And I'm like, it looks, they say it looks like, stacked, flattened pancakes. Maybe, if they're green. And granted, they just make them look green on the picture, but it has a whole bunch of little stacked folds. Now, the main job of the Golgi apparatus is things are going to go to it. It is a receiving center. So things are going to go there. It's going to modify those things. A lot of them is proteins. It can concentrate it. It can change them up a little bit, repackage them, and then ships them off. So a lot of things are coming from the rough endoplasmic reticulum. And these little vesicles that come from the rough endoplasmic reticulum. And they're going to fuse with the Golgi apparatus. I shortened it. They get modified, they get tagged for delivery, they get sorted, they get packaged, and then they get shipped out. I like to think of the Golgi apparatus as a distribution center. Things are going to go in there, they're going to arrive, they get repackaged up, they get reorganized, and then they get shipped off to wherever they need to go. So this is showing that a little rough endoplasmic reticulum vesicle with some proteins in it is going to arrive at the Golgi apparatus. It's going to fuse. All those proteins are going to go in there, they're going to get sorted, they're going to get repackaged, and then they're going to get shipped out in their own vesicle to go wherever they need to go. Maybe it's for some type of secretion out of the cell, maybe it's to go to some other type of organelle, but they get shipped out. Now, an actual picture of the Golgi apparatus is this one. It's magnified 90,000 times its normal size, but you can see all those stacked folds. You can even see some of the vesicles that are forming there. Now, this is just to kind of give you a basic, idea of the rough ER working together with the Golgi apparatus. Things are leaving the rough endoplasmic reticulum, fusing with the Golgi apparatus. They get sorted, repackaged, and then shipped out, whether it's for secretion or to become some other organelle like a lysosome. They're working together. Now, some of these lysosomes, some of these vesicles that leave the Golgi apparatus can actually become a whole nother organelle itself. It really depends what's inside of there. because a lot of them are proteins. Well, these little blue dots could be enzymes. Enzymes are proteins. And the main job of a lysosome is to take these enzymes, and the enzymes break things down. And so a lysosome is a membranous, they call it a membranous bag. And I'm like, it's a membranous organelle that has digestive enzymes. And this is an area where things are broken down. It's called intracellular digestion. Those enzymes are going to break apart any bacteria, viruses, and toxins that certain cells have ingested. They can break down any organelles that aren't working correctly. They can break down glycogens if you need energy sources. They can break down non-useful tissue that if you injured it and it's dying and we don't need it anymore. And if you need calcium in your body to work with other chemical reactions in the body, they can actually break down bone to release needed calcium if you're not getting enough calcium in your diet. So their main job is to break things down. I like to think of them as the cleaner uppers of the cell, like Lysol cleans things up. I'm like, this really helps clean things up. It gets rid of things that shouldn't be there, bacteria, virus, toxins. It gets rid of, organelles that shouldn't be there. It gets rid of tissue that shouldn't be there. It's kind of the cleaner upper of the body. And this is an actual picture, big orange blobs of lysosomes, and the green areas are different places where materials, whether it's an organelle, whether it's a bacteria or something that was ingested, they're being digested by those digestive enzymes. Another part of the cell is the cytoskeleton. Now, just like we have a skeleton, our bones, and I'm like, to help give us structure, so does a cell. So it's a network of rods, kind of like our bones are a network of rods, and I'm like, that run through the cytosol, and there are lots and lots of different things that hold it all together. And their main job is to become the bones. So it can help move the cell can change shape a little bit, and because it has all these little interlinking bones on the inside, it actually allows various organelles to move around inside the cell. They can link onto one of these cytoskeleton rods and then move along it, so they can go from point A in the cell to point B in the cell by following one of these little rod structures. So things can move around inside the cell if it needs to, but it also provides the cell some shape and structure. Centrioles, it's usually located near the nucleus. Centro means the center, so it's located near the center of the cell. And its main job, because there's usually pairs of them. I always think they look like little Twizzlers, but yellow in the picture. Their main job, is little barrel-shaped or microtubule organelles. Their main job, because we'll get it into with mitosis, is they really do help with cell division. Some of their main job is to send out little spindles to help break apart the chromosomes during cell division, and because of their structure, those little tube-like structures, they can form the basis of cilia and flagella if the cell has it as well. Now, the cilia and the flagella. Not every cell has them, but they're whip-like modal extensions on the surfaces of certain cells, not all cells. They contain microtubules. You can see all of those little tubes, very reminiscent of these exact same tubes because they form the basis of them. And the function of cilia is to move stuff across the surface of the cell. So here's our ciliated cells in here. Here's some mucus, and these whip-like cilia are going to do just that. They're going to whip in kind of a beating fashion to move that mucus across the cell. We find cells in the body that have these cilia to do that in the respiratory tract. So you've got mucus that's trapping dust particles, and we're trying to get rid of those dust particles out of the respiratory tract. It's kind of a cleaning area of our cells. Now, that means we don't have any cells in our body that move around with cilia. There are single-celled organisms in the world that are covered in cilia and use the cilia for movement. These cells themselves they're stuck in place. They are not moving. They just use the cilia to move stuff across the cell. The flagella, if a cell does have a flagella, and the only cell in the human body that can have a flagella is a sperm, its main job is to move the entire cell, the whole sperm. And it works in a whip-like fashion and moves that entire cell. Now, if a cell has microvilli, another cellular extension, They look like little finger-like extensions. And everyone's like, ah, so they look like cilia. A little bit. And I'm like, big difference though is these don't move back and forth. Their main job is just to increase surface area. So I'm like, they're not beating back and forth. They're not moving anything across it. They just have lots and lots of surface area. We find cells that have these on them in the small intestine because there's lots and lots of absorption in the small intestine, and the more surface area, the more absorption you can have. It's not for movement, just absorption. Now, biggest organelle we have inside the cell is the nucleus. The largest organelle. It's massive inside there. And its main job is to house the DNA. It has the entire genetic library to make any protein it wants. And again, we find proteins everywhere. It's the one that determines what kinds of proteins we need to get that we need to have made, and how many of those proteins. Now, most cells are uninucleate, which means they have just one nucleus. Most cells do. However, there are a few that have more than one. Some skeletal muscle cells have more than one nucleus. Some bone destruction, breaking down cells have more than one nucleus. And some liver cells are multinucleated. Most in the body just have one, but some have more. And one unique type of cell doesn't even have a nucleus. Red blood cells are anucleate, meaning they're missing a nucleus. At one point in their life cycle, they did have one, but then they lose it, which gives the red blood cell shape kind of a flattened donut look. So they lost that really big organelle. Now, here's the size. You can see size of the cell, nucleus, biggest organelle inside of there. Now, on the outside of the nucleus, it does have a double membrane. It's got two white layers there. The outer is connected with the rough endoplasmic reticulum that has all those ribosomes on it, and the inner one maintains the shape of the nucleus itself. So one is attached to the rough ER, one just maintains the shape of the nucleus itself. And it does have pores. It has little tiny holes that allow things to go inside the nucleus and allows things to come outside the nucleus. Now, one structure inside the nucleus is this blue structure in there that's called the nucleolus, or nucleoli for plural. They're dark staining spherical bodies inside of there, and the main job of the nucleolus in there is to make ribosomal RNA and to help assemble ribosomes. And again, the function of a ribosome is to make proteins, but they had to come from somewhere. And those ribosomes really are coming from inside the nucleus, from that organelle called the nucleolus. Now, again, one of the biggest things found inside the nucleus is DNA, and DNA is organized into threads called chromatin. So chromatin is made up of DNA, but to organize all of that DNA into these big, huge threads that we can even see underneath the microscope, and we have a bunch of proteins that these DNA can wrap around. So we've got DNA that's wrapping around proteins that help keep that DNA organized, and we're also going to find some RNA. Now, when this DNA wraps around the proteins, they are called little fundamental units. They're called little nucleosomes. It's just some of this DNA, a little bit of RNA, wrapped around these histone proteins. These histones just pack long molecules of DNA that are involved in maintaining certain types of genes that are coding for making proteins. Again, in one cell in your body, just one cell in your body, you have 6 feet of DNA. It's really skinny, really, really skinny. So you have lots and lots of DNA to help keep that all organized. And we wrap it around these proteins and package it into these little fundamental units called nucleosomes. Now, when you condense all of that 6 feet of DNA mixed and wrapped around these proteins for organization, when it shrinks all down, it does show up as little X-like structures called chromosomes. And so when all of that DNA condenses, we see it almost as little X's. That all of that DNA forms these X-like patterns, and those are the chromosomes that we're going to split when we go through mitosis. Now, little review of this little section, part C, of all the organelles. So what is the fluid component between the plasma membrane and the nuclear envelope? And the fluid part, just the fluid, is the cytosol. The cytoplasm is all of that. It's The cytoplasm is the cytosol, the organelles, and the inclusions. Just the fluid part is the cytosol. You would expect that cells that expend a great deal of energy, such as skeletal muscle cells, would have an increased quantity. And if you need lots of energy, you're going to want lots of mitochondria that can make lots and lots of ATP. The following cell that does not have a nucleus when it's mature, it's anucleate, are your red blood cells. And I was going to say, and that's my last part of part C. We'll continue part D in another part.